Picking up where I left off on the last lecture, we will be moving into the mid and hindbrain, starting with the basal ganglia, which is this weird looking thing right here. So the basal ganglia, it's still technically the forebrain, structures in the forebrain that help control movement and motor planning. Damage to this section can actually contribute to Parkinson's disease. And Tourette's syndrome, which is characterized by both motor and vocal tics, seems to be associated with abnormalities in this region as well. And then the limbic system is the emotional center of the brain that also plays roles in smell, motivation, and memory. So this processes information based on our internal states like blood pressure, heart rate, etc., as well as our emotions. This region evolved out of the primitive olfactory system, which controlled various survival behaviors in early mammals. This is part of the reason that smell can elicit such strong memories. Like, for example, if you get food poisoning following eating something in particular, it's actually a survival mechanism for you to never want to eat that ever again because it made you so sick. And sometimes even the smell of it can be nauseating. And this region has 5 main areas. We have the cingulate cortex, which is not shown here, the thalamus, the hypothalamus, the amygdala, and the hippocampus. So the cingulate cortex is the primary cortical component of the limbic system, which is involved in emotional and cognitive processing. The thalamus is the gateway from the sense organs to the primary sensory cortex. Most sensory info passes through here and undergoes some initial processing before moving to the cortex. The hypothalamus is the part of the brain responsible for maintaining a constant internal state, or the biological term is homeostasis. That is the hypothalamus's job. And different areas here play various different roles in emotion and motivation, some for hunger, thirst, etc. This is really known for the 4 F's. Feeding, fighting, fleeing, and sexual activity. So I'll let you guys fill that last one in for me. It also helps regulate body temperature. The amygdala is the part of the limbic system that plays key roles in fear, excitement, and arousal. It also plays a role in fear conditioning as well. So as per the ethics lecture, if you guys remember little Albert, this is the part of the brain that was responsible for that effect. And the hippocampus is the part of the brain that plays a role in spatial memory. So right now, all of you can probably see, like, okay, you have your house, and looking at it from the outside, you can pinpoint where in the house your bedroom is. Or if you live in an apartment, you can look at the building from the outside and pinpoint where in that building your apartment is. And that's spatial memory. It also lets you know, or helps you remember where certain corners are so you don't constantly stub your pinky toes and stuff like that. So when we make a map of how we get from one place to another, that's using our hippocampus. This area is actually found to be larger in taxi drivers than in non-taxi drivers, and larger in experienced taxi drivers than in newer taxi drivers. And damage to this area leads to difficulties forming new memories, but leaves old memories intact. This is part of the reason elderly people with Alzheimer's or dementia tend to retrace old steps. One hypothesis is that the hippocampus houses memories temporarily before transferring them to other sites, such as the cortex for permanent storage. Another is called multiple trace theory, which believes memories are stored in multiple sites and over time. Some sites get stronger and others get weaker, therefore memories aren't necessarily transferred, they're already there. So this is the hind and midbrain. We're going to be talking about this portion right here, the cerebellum, this weird yellow looking area. It is responsible for our sense of balance. So this is the mini version of the cortex. It's part of the hindbrain. It plays a predominant role in senses of balance and enables us to coordinate movement and learn motor skills. Damage to this region typically results in severe balance issues. It also contributes to executive, memory, spatial, and linguistic abilities. Next, we have the brainstem, which is this green area right here. And this is the part of the brain between the spinal cord and the cerebral cortex that contains the midbrain, pons, and medulla. This performs some of the basic bodily functions that keep us alive. So the midbrain plays an important role in movement and controls the tracking of visual stimuli and reflexes triggered by sound. So it contains the reticular activating system, called or abbreviated as the RAS, brain activity that plays a key role in arousal. This connects with the forebrain, the cerebral cortex. Damage to this area can result in a coma. Pathways from the RAS activate the cortex by jacking up the signal-to-noise ratio among neurons in the brain. So signal-to-noise ratio, think of it as you're talking on your cell phone and you start getting static. That's more noise than there is signal. But if there's no static or very little static, then there's more signal than there is noise. And so pathways from the RAS increase the amount of signal and decrease the amount of noise. As far as the pons and medulla, these are regions of the hindbrain, which is the region below the midbrain. So right here, here's the pons, here's the medulla. And this contains the cerebellum. Oh no. The pons does not contain the cerebellum. I misread my notes. Disregard that. The pons plays a crucial role in dreams and connects the cortex with the cerebellum. The medulla is the part of the brainstem involved in basic functions like heartbeat and breathing, which is why some people feel like they're going to throw up after a bonk to the back of the head. Serious damage to the medulla can cause brain death, which is an irreversible coma. And the spinal cord is the thick bundle of nerves that convey signals between the brain and body. Nerves extend from the spinal cord and run all throughout the body. Sensory info is carried by sensory nerves, and motor information is carried by motor nerves. This also contains interneurons, which sends information to nearby neurons. These connect motor and sensory neurons without having to report back to the brain, and the result is called reflexes. This, of course, is an evolutionary advantage. If you touch something hot, your body needs to react quickly. You don't want to get your brain involved per se. So you touch hot, pull back, that's a reflex, and the interneurons are responsible for that. Next, we have the peripheral nervous system. And this is comprised of the nerves leading out of the spinal cord. So it has two different sections. As I mentioned earlier, we have the somatic nervous system, which carries messages from the central nervous system to muscles throughout the body, controlling movement. And whenever we use our joints to stabilize our body, the CNS and the PNS cooperate to regulate our posture and movement. And then the autonomic nervous system controls involuntary actions of our internal organs and glands, which, along with the limbic system, participates in emotional regulation. This includes breathing, heartbeat, salivation, etc. The brain, especially the limbic system, interact with the autonomic nervous system to control emotion and physical states. And the autonomic nervous system has two subsystems, the sympathetic and the parasympathetic. So the sympathetic nervous system gets engaged during a crisis or after actions requiring fight or flight. So this is our survival nervous system. We see something we perceive as a threat, the sympathetic kicks on. It's like, okay, action is needed right now. And we've all heard of the fight or flight responses. There's actually a third called freeze, which is probably an odd evolutionary adaptation resulting from some predators losing interest in their prey when they stop moving. And then I couldn't help but think of Jurassic Park when I was reading through this, the scene with the T-Rex, but I couldn't find a good screenshot of that with the subtitles, so I have this little meme here for y'all with Barney and the Jurassic Park people as little kids. And then sympathetic activation triggers a variety of responses that help in a crisis, like heart rate and blood pressure. And the parasympathetic nervous system controls rest and digestion, and this kicks into gear when a threat is no longer on our radar. So this is more the resting state nervous system. So the sympathetic is the gotta go, need to engage now nervous system, and the parasympathetic is like, whoa, slow down, man, no need for all of that nervous system. Next, we have the endocrine system, which is a system of glands and hormones that controls secretion of blood-borne chemical messages. And hormones are chemicals released into the bloodstream that influence particular organs and glands. So first is the pituitary, which is located in our brain. The pituitary gland is a master gland that, under the control of the hypothalamus, directs other glands of the body. So again, another business analogy for you guys. The pituitary gland is the CEO or the board of directors, and the rest of the glands are the other positions or departments in the company. The pituitary releases a variety of hormones that serve numerous functions, ranging from regulating physical growth, controlling blood pressure, and determining how much water we retain in our kidneys. Oxytocin is one of the main pituitary hormones and has gotten a lot of attention in recent years. It's responsible for multiple reproductive functions. It's also a bonding hormone, and that it plays a major role in both maternal and romantic love. It also seems to affect how we trust others. It's because of oxytocin that we tend to treat those close to us better than we do strangers. And this is believed to be caused by oxytocin increasing our sensitivity to social cues. Next, we have the adrenal glands, which are right here. Adrenal gland is a tissue located on the top of the kidney and releases adrenaline and cortisol during states of emotional arousal. Adrenaline boosts energy production in muscle cells, thrusting them into action while conserving as much energy as possible. Adrenaline triggers many actions, including contraction of our heart muscle and constriction of our blood vessels to provide more blood to the body. It opens the bronchioles, which are the tiny airways in your lungs, to allow more inhalation of more air. The breakdown of fat into fatty acids, providing us with more fuel to run toward potential prey or away from potential predators. The breakdown of glycogen, which is essentially carbs, into glucose, sugar, to energize muscles. And then the opening of the pupils of our eyes so we can better see during emergencies. And adrenaline allows people to perform amazing feats. I'm sure you've probably all heard the story of how the mom was able to lift a car off of her child, and that was due to adrenaline. And cortisol increases our response to physical and psychological stressors. So, for example, in some anxiety disorders, people have elevated cortisol levels even in their resting state, and that's because of the anxiety. And then as far as sexual reproductive glands and sex hormones, of course, Bio 101, testes, ovaries. Of course, the testes produce testosterone, the ovaries produce estrogen and progesterone. However, women still have testosterone because both the ovaries and the adrenal glands both produce low levels of testosterone. Mapping the mind. All right, so this is, I don't want to say it's a relatively new field, but the advances we've made in this area are relatively new. So one of the first techniques was phrenology, which was just fundamentally flawed all around. So if you look at the diagram I have for you guys right here, this is essentially what phrenologists believed. They thought bumps on the head corresponded to enlargements or detriments in certain areas of the brain. And if that sounds like a load of crock, it is. But it was really popular during the 1800s, so you have to give them a little bit of a break, because it was the 1800s. They didn't even have cars yet. And this was one of the first attempts to map the mind onto the brain. So phrenologists assumed that enlargements of the skull corresponded to brain enlargements, and that those enlargements were directly tied to psychological capabilities. And of course, the white Anglo men used the practice of phrenology to try and demonstrate racial and gender superiority. This is actually mentioned in a scene from Django Unchained, and unfortunately that was a very common belief of the time. While it was wildly inaccurate, it was used to validate systems of oppression and keep those systems of oppression in place. And as if that's not bad enough, it later fueled a practice called eugenics. And for those of you that know what that is, you know how horrible it was. And for those of you not familiar, I encourage you to look it up, just because it kind of goes outside the scope of this class, especially this lecture. But it has a dark history all its own, and yeah. But, you know, not surprisingly, phrenology was not only inaccurate, but they weren't even measuring the bumps on the brain like they thought they were. They were literally just measuring the skull. Because the shape of the brain is not a one-for-one match to the shape of the skull. But again, 1800s, they couldn't exactly just do voluntary brain surgery. So they were kind of operating within the realms of what they could do. And as far as brain damage, so new methods developed to study psychological functioning following damage to specific regions of the brain. These methods included lesions in areas of the brain to study the resulting behavior and function. So originally this was done in small animals. Today's neuropsychologists don't really use lesions anymore. They use, advanced imaging and psychological tests that don't involve brain surgery or damaging the brain of their subject. Because, shockingly, it's not exactly ethical to give your patients brain damage. With that being said, sometimes they will study individuals who already have lesions. So again, those who have had strokes, advanced Alzheimer's, dementia, subjects like that, or traumatic brain injuries, they will study those participants, but they are not inducing the damage. They are merely observing the damage after the fact. So brain scans. This is really what most people use now to observe the brain and study it in more detail. So researchers soon discovered that by stimulating certain parts of the brain during surgery caused extremely specific movements, like specific enough to where they could poke a spot to twitch your index finger and poke a different spot to twitch your pinky finger. This led to the idea that neurons use electrical activity to send information to one another. And based on this idea, the electroencephalograph, or the EEG, which is this one, was developed to measure electrical activity in the brain. This allows scientists to determine if a person is awake or asleep. So, since this is non-invasive, it's pretty common in research, especially sleep research. It can also detect rapid changes. It's commonly used to study patients with schizophrenia, epilepsy, and other disorders. These scans aren't the best at determining which area of the brain is active, since it only captures the electrical activity that reaches the scalp. So if you look at this picture, this is what an EEG looks like. And as you can see, it's only the scalp, it's not going any deeper, and it only captures the electrical activity that reaches the scalp, which is why it makes it difficult to determine which brain regions are active and which are not. So, other techniques include the computed tomography, or the CT scan. And this is a technique using multiple X-rays to construct three-dimensional images. Next, we have the magnetic resonance imaging, also known as MRI, which uses magnetic fields to indirectly visualize brain structure. And this measures the release of water from biological tissues following exposure to a magnetic field. So both of these techniques allow us to visualize the brain structure. Both display a lot more detail than a single X-ray, but both only show structure, not activity. Which brings us to the positron emission tomography, or the PET scan. And this measures consumption of glucose-like molecules, yielding a picture of neural activity in different regions of the brain. And they use glucose because they believe that neurons, like other cells, consume sugar when they activate. So this is essentially measuring how the brain responds to a certain stimulus. This scan requires the injection of radioactive molecules to trace the activity. However, these are short-lived, so there's little to no harm done to a patient. I don't know if any of you have ever had to have like an upper digestive tract scan, but it's basically the same thing. Same with gallbladder test. They give you just a little bit of radioactive dye, wait for it to kick in, and then it goes away after, not too long after the scan is finished. But because they do have to inject radioactive dye, this is considered an invasive procedure, which is why scientists went on to develop the fMRI, or the functional magnetic resonance imaging. And this uses a magnetic field to visualize brain activity using changes in blood oxygen level. So they use the blood oxygen because they believe that, like other cells, neurons consume oxygen when they activate. This is now the standard functional imaging technique. And it's commonly used to visualize brain activity in response to specific tasks. And then next we have the transcranial magnetic stimulation, which is TMS. This is a technique that applies strong and quickly changed magnetic fields to the surface of the skull that can either enhance or interrupt brain function. This technique provides useful insights as to which brain areas are involved in different psychological processes, and this is the only non-invasive imaging technique that allows us to infer causation. The rest only give us correlational data. And lastly, we have magnetoencephalography, the MEG or MEG, and this is a technique that measures brain activity by detecting tiny magnetic fields by the brain, or generated by the brain. The major strength here is the ability to track brain changes over extremely short time intervals. Now, while all these scans are well and good, there is still a lot of misinterpretation that goes on with the results. So many people assume that functional images like fMRIs and PET scans are essentially photos of the brain in action when they actually aren't. In most cases, the images are produced by subtracting the brain activity on a controlled task from the brain activity on an experimental task. And researchers also typically condense findings across individuals as well. So for example, they use group level data like means, averages, stuff like that, instead of like individual data. And lastly, these scans do not allow us to read minds. I don't know where this myth started. It's ridiculous, but it's not true, not even a little bit. So, how much of our brain do we use? So, as addressed in the Chapter 1 lecture, there is a myth that people only use 10% of our brain, which is absurd. This is a total myth and is entirely inaccurate. Uh, However, the early inability to properly measure brain activity didn't really help here. It just furthered this myth. And then the parts of our brains, what do we use for what? So a lot of media oversimplifies this. As we saw in this chapter, there is a lot of overlap between various areas of the brain when it comes to functionality. So yes, while some areas tend to focus more on certain things, like the limbic system focuses on emotion, it still influences other systems, and it contributes to their function as well. So this is a hotly debated subject on all social sciences. So nature versus nurture. Who hurt you more, your parents or your genetics? So biological heredity. We know that people, plants, and animals all have chromosomes, which are slender threads inside a cell's nucleus that carries genes. Genes are genetic material composed of DNA. And one thing I did want to mention here is the Human Genome Project is super cool. Highly recommend everyone check it out. Go to their website. I don't know if you'll be able to access the URL I provided here, but if not, you can just go to Google, type in Human Genome Project. Because this was completed in 2003 and characterized all human genes, all of them. So this next portion is directly from their website. The Human Genome Project has revealed that there are probably about 2,500 human genes. This ultimate product of the Human Genome Project has given the world a resource of detailed information about the structure, organization, and function of the complete set of human genes. This information can be thought of as the basic set of inheritable instructions for the development and function of a human being. And again, a lot more information can be found on their website. I just wanted to include this little snippet. So, as far as genes, there are what's called a genotype and a phenotype. So a genotype is your genetic makeup. So for example, having XX chromosomes. Your phenotype is the observable trait, which would, in most cases, be being a woman. And one thing I do want to include here is I do say most cases because there's a thing called intersex, which is where you may be biologically a woman with the reproductive system of a woman but not have XX chromosomes. And there's a lot of different variations that can go on there. It is a very broad umbrella term. So if you're interested, I would suggest giving it a quick Google. So yeah. And then genes can also either be dominant or recessive. So a dominant gene masks the effect of other genes. And a recessive gene is suppressed by a dominant gene, therefore it's only expressed when there is a lack of a dominant gene. So for example, brown eyes are a dominant trait. So people with blue or green eyes will only have blue or green eyes in the absence of the brown eyes trait. And as far as behavioral adaptation, so behavioral adaptations are changes in either genetic makeup or behavior. Successful adaptations are changes that are better suited for the environment in which an individual lives. Individuals who possess these successful adaptations are more likely to survive and procreate than those that do not. Successful adaptations increase an organism's fitness, which is the capacity to pass on their genes. So evolutionary psychology, a field I personally believe should have more classes offered at a lot of schools, tries to explain psychological traits as naturally selected adaptations. Honestly, nature kind of dropped the ball on mental illnesses, especially depression and anxiety, but I digress. As far as the human brain evolution, the relationship between our brains and our bodies has been finely tuned over millions of years, which is why our behaviors are more complex than most other mammals. Now, with that being said, there are exceptions. Higher apes can learn sign language, dolphins are highly empathetic, etc. This is just in general our behaviors are more complex. And then lastly, behavioral genetics. So scientists use an approach called behavioral genetics to examine the influence of nature and nurture on psychological traits like intelligence. This approach looks at genes and environmental influences. This allows us to estimate the heritability of traits and diseases. Some traits are heritable, some are not. Height is heritable, but a religious affiliation is not. There are a couple different behavior genetic study designs. So scientists estimate heritability by using one or more of 3 behavioral genetic designs. There are family studies, twin studies, and adoption studies. So a family study is an analysis of how characteristics run in intact families, and the field uses the term intact families to mean all family members live in the same house. So biological mom, biological dad, biological children. That would be an intact family. This info can be used for estimating risk of a disorder among the relatives of people afflicted with that disorder. There is twin studies, which is the analysis of how traits differ in identical versus fraternal twins. Now, I know I mentioned the horrible twin studies done by the Nazis in my last lecture. Those are absolutely not the same as these twin studies, not even close, so please do not conflate the two. The twin studies I mentioned here are done well within the bounds of the ethics code. The studies designs are approved by an institutional review board. Like, they pass every checkpoint along the ethics line. So I just really want to emphasize that. So twins can either be fraternal, which is from 2 different eggs, or identical, which is when a single egg splits into 2 zygotes. And so those twins have nearly identical DNA. And this helps differentiate which effects both nature and nurture have on people. So if people have very similar DNA, but experience the same or similar upbringing, this helps us separate the effects of environment versus DNA. And then you have adoption studies, which is the analysis of how traits vary in individuals raised apart from their biological relatives. One potential confound here is the selective placement of children into adoptive homes, because adoption agencies most likely won't put a kid in a home they deem to be potentially or actually harmful. Like, the adoption process is a lengthy process. There are a lot of things adoptive parents or potentially adoptive parents have to do in order to be approved to receive an adoptive child. And then lastly, there is epigenetics, which is a field that focuses or that examines how environmental influences affect the expression of genes. So the most common examples I can think of here are depression and anxiety. Both of those are heritable disorders. There is a gene for depression and anxiety, or gene combinations. I can't quite remember exactly, but the expression depends on the environment in which the child is raised. So if they grow up in an unstable environment, they are more likely to have depression and anxiety expressed than a child that grows up in a very stable, structured environment. So just a couple quick housekeeping things. Remember, quizzes for chapters 1, 2, 3, and 5 are due Saturday, February 20th at 9 p.m. Central Standard Time, and Exam 1 is due Sunday, February 21st, 9 p.m. Central Standard Time. And again, if something comes up and you think you might miss the exam, even if you don't end up missing it, if you think you might, you have to let me know beforehand. I can help beforehand, I can't help after the fact.